Today we're going to talk about the basics of how to write a grant proposal. So this is going to focus on workshopping a really basic project proposal, something that might be written for an undergrad project proposal or a very early graduate research grant. So to get started, let's talk about the basic elements involved in any proposal. So before we go into the details, I just want to focus on the fact that I have suggested lengths here, but that these will likely change according to the overall length of the proposal. So this is sort of with a 2-page proposal in mind, but a lot of grants are about 6 pages or 12 pages, and so you'll want to adjust accordingly. So the first section in any proposal is going to be your introduction or your background. This is going to be usually 1 paragraph or half a paragraph that goes over the basic topic or disease of interest, and then 1 or multiple paragraphs for each new technology or gene or drug that you're going to be talking about in your proposal. This is also your place to put in lots of references to show that you know what you're talking about and you've done your background research and you're really aware of what's going on in the field and what's going on with your specific topic of interest. So, like I said, this should include background on the disease or on the topic, and then any background on anything else that you're going to introduce in your proposal that's new. The next section, which isn't really its own section, it's usually put at the end of the introduction, is your hypothesis or problem statement. So this should really clearly highlight the gap in knowledge. So what is it that's unmet or that people don't know about that your proposal is going to address? It should talk about any existing data on the topic. So this, again, should be very well referenced to show that you know what exists in the field and what's possible. And then it should address the research question in a very clear statement that says, this is what I'm going to do, this is my hypothesis, and this is the potential impact of that project. This is really like one of the strongest statements in your proposal, so you want to be very clear about what it is you're going to do and why it matters in this sentence. Next, for some grants, you're going to include preliminary data. So this is 1 to 2 paragraphs based on how much you have. You should really only include this if it's going to be additive and helpful. So if it's something really interesting or cool or shows that that your project is definitely going to work, then you want to make sure to include that. Whatever you do include, you should describe very clearly, and any figures you add should be very well labeled and referenced in the text so that people know what that figure is important for. The next piece of your project is going to be the project description. So this is usually subdivided into 2 to 3 specific aims, and you can think of aims as like sequential pieces of the project. So it should be, first I need to do this, then I need to do this other thing, and then I need to do this third thing in order to achieve this entire project. So, and each aim should have at least 1 very detailed paragraph, usually more for longer grants. So for each aim, you want to talk about the main question that this aim is addressing and how that question contributes to the overall project. You also want to talk about the methods you're going to use, so detail each experiment and what you're going to do. And remember that every single experiment you propose should have a clear value add to the project. You don't want to propose extraneous things or things that don't really follow logically. Make it very clear how each experiment is helpful. Then you want to talk about the expected results from those experiments, and you want to explain how they will contribute to your project if they work. And finally, you want to briefly address the expected timeline for this aim. At the end, you want to talk about significance. So this is usually a pretty succinct paragraph, but it should be very well written and hard-hitting, and it should really answer the question of why someone should care about your project. So this is where you tell them why your project is important, why it's novel, why it's something that they should fund, why it's interesting. So this should be, again, like a part of it that is very strong and very well written to show that your project is important. Most grants also include a bunch of attachments, and so the first and major attachment that will be in any grant is your reference list, and it's good to reference your grants well. So take the time to look up the relevant papers and reference everything to show that you did your homework and you know what you're talking about and you know what's out there in the field. That really convinces people that your project should be funded because you do know what you're doing and it's likely to work. So list your references in accepted and consistent format and reference really well good places to include references are your introduction, your background, and then when you talk about existing data in the field. And then for every grant, there's usually other documents involved, so you should check the requirements carefully. Often these are letters of recommendation, a CV, sometimes an institutional profile, so make sure you check all of those requirements as well. Okay, so today we're going to workshop an example project. So in this project, we want to run a CRISPR screen to identify genes and pathways that contribute to resistance to Avastin, which is a drug, in GBM, which is our disease of interest. So we're going to walk through a very simple proposal, walk through a few examples for this particular question. Okay, so let's start with our introduction. So we already talked about how you need to make sure to introduce every single component of your project one by one. So for the disease, our disease of interest is going to be GBM. And then a few things that you might want to say about GBM are that it is the most common malignant brain tumor in adults. That tells people why it's interesting to study. You might say that it's derived from astrocytes, that it's highly aggressive, but the most important thing to mention is the standard of care and the fact that there is a 21-month average survival and a 100% fatality rate. Those are all great reasons why you'd want to study this disease and why you'd want to do projects on it and find new targets. And so this really helps you sell your project. The next thing you're going to want to talk about is the drug, because that's another piece of our project that we're introducing. So Avastin is a drug that's normally used to inhibit blood vessel formation. It's used in many diseases, and it was recently approved by the FDA to treat recurrent GBM. It's useful, or it's thought to be useful, because tumors often form their own blood vessels, and that allows them to become more aggressive because they can deliver nutrients to the tumor. And so it was thought that Avastin would be helpful. Clinical trials in GBM have shown that it does increase progression-free survival, which means that patients have longer time periods without symptoms, but it does not improve overall survival. And people think that this is likely due to the development of resistance to Avastin over time. So like we've talked about previously, the cells evolve over time and become resistant to various drugs, and Avastin is one of them. And then finally, we want to provide a background on the technology we're using. So this is CRISPR-Cas9 and the concept of doing a large-scale screen with CRISPR-Cas9. So some things we might include here are saying that CRISPR-Cas9 is a way of introducing DNA-level knockouts. We want to say what it is. We also want to say how it works. So here we've talked about the guide RNA and the Cas9, so this is how it works. And then you want to talk a little about screens. So why is CRISPR-Cas9 good for large-scale screens? And some of those reasons are that it is efficient, or that it's very quick to use, it's easy to use. It's easy to multiplex. You can put multiple knockouts into a group of cells. So all of those things allow us to use it as a large-scale screening method. And finally, the thing you want to mention about CRISPR-Cas9 is that it allows us to understand functions of genes. And our goal here is to really understand which genes function as resistance genes. And so we can look at the whole genome or a large set of genes, and CRISPR-Cas9 screening allows us to identify those functions. And that's why it's a useful technology here for this kind of question. So altogether, we've introduced all the major components of what we're going to talk about in the future. And that's really what your introduction should do, is it should give the reader a good framework for how to move forward and understand the rest of your grant. And it should also really set up your question. And so for the person to understand our question, they need to understand all of these things. They need to understand how bad GBM is, and they need to understand that Avastin doesn't work. And then they need to understand the screening technology we're going to use. And it's only then that we can set up our actual question and hypothesis of what we're going to do. Okay, so this segment is just the introduction to the disease. So this is just meant to be talking about GBM, the piece that is an intro to GBM. So let's go through this. So the first thing is that GBM is the most common type of aggressive astrocytoma in adults. So here I probably wouldn't say astrocytoma, because that's another word that the reader might not understand. So you've explained one word they don't understand. with another word that they don't understand. So instead of saying astrocytoma, I would probably say is the most common primary malignant brain tumor in adults. And the reason we say primary malignant brain tumor is because you need to make sure to include all of those specifications. If you just said primary brain tumor, it wouldn't actually be true, because the most common primary brain tumor would be a meningioma. So you have to make sure to say primary malignant brain tumor in adults, and that's how you make it a completely true statement. The next piece they've said is that it forms from glial cells in the brain. So this is something that, although it's interesting and perhaps helpful, is not the most necessary when we're just talking about GBM and Avastin. We really want to focus on the fact that it is a common tumor and that it affects people badly. We don't really need to get into all the details of, what it forms from and what age groups it affects and so on. We just want to provide the relevant information. So according to that, I would probably also cut this later sentence where they say it has a late onset and affects certain age groups, and has slightly higher occurrence in men over women, because all of those things, while they're interesting and it's good that the person has researched them, it's maybe not the most helpful information in terms of setting up just why we want to study GBM. This piece here is actually a really interesting piece, where we talk about the blood supply and the uninterrupted tissue growth, because we're later talking about Avastin. And as we've already discussed, Avastin does relate to the blood supply. So although I would not normally introduce this part in a general grant because it's not the most relevant, I do think it's helpful here because we're talking about Avastin later. Okay, so moving past that, now we move on to therapy. So when this person says aggressive therapies, I would actually expand on that. It's important to say exactly what those therapies are. So it's important to say that we use TMZ-based chemotherapy, we use radiation, and we use tumor treating fields and surgical resection. And that all of these things together result in a survival time of 14.6 months. This has now been updated to 21 months. And with less than 5% of the patients surviving more than 5 years. So this is a really bad prognosis for any cancer. And so it's important to stress that, and it's important to stress how many aggressive therapies we're giving and how the prognosis is still really bad. So you want to make sure to mention that at the beginning of any proposal you write, because it really justifies for you how important it is to study this disease. So it gives your grant an automatic justification in a way. And then this sentence here, chemotherapy drugs are usually effective due to the ability to cross the blood-brain barrier. So this is not exactly what we want to say. It is true that chemotherapy drugs are initially effective, but it's not because they cross the blood-brain barrier. That's a given. For any drug that should be effective, it should cross the blood-brain barrier. The reason that they're initially effective is because the cells are not resistant yet. So what you actually want to say is that chemotherapy is initially effective, but then the effectiveness goes away because of resistance. So then the effectiveness is going to go down, and it's going to be because of resistance. And that's how you set up, well, we need therapies for resistant tumors, and that's why we look at Avastin. So overall in this introduction, the things we really want to highlight are what GBM is. We might talk a little about the blood supply because it's a nice tie-in. And then we want to focus really on the current standard of care and the survival issues and the resistance, because that sets us up well for justifying why we want to study this disease, and also sets us up well for talking about Avastin later on. So this is an edited version of this paragraph with all the edits we just discussed. So as I said, you want to make sure you say primary malignant brain tumor because that's what makes it true. You want to make sure you use words people know to explain the words they don't know. This is a good tie-in. And this is unnecessary, all of this. You don't really need to get into it. You do want to say the exact current standard of care, and you also want to say the exact survival time, because that justifies why you're studying it. And then you want to explain that it's because of recurrence and because of resistance. And that's why we need new therapies. And so this is a really nice transition into your next paragraph or your next piece of background. Okay, so now let's talk about the next piece of the introduction, which is the introduction to Avastin, which is our drug of interest here. So we already set ourselves up well by saying that GBM is resistant. And so now we can transition neatly into talking about new drugs for resistant GBM. So this is not the correct year. It was much earlier. It was in the 2007-2009 range. A new drug was approved by the FDA to treat recurrent GBM. So this is good. It's a good intro. And this person mentioned the generic name, which is important to mention when you're talking about a drug. Then they talked about how GBM causes breakdown of the blood-brain barrier and extracellular cerebral edema. But I think although it is true that GBM causes breakdown of the blood-brain barrier and does cause edema, the more important thing here is that GBM makes its own blood vessels, and we want to prevent it from doing that. Because when it makes its own blood vessels, it's able to deliver more nutrients to the tumors, and so that allows the tumor to grow more than it normally would. So although it is important that it messes with the blood-brain barrier, it's more important that it makes its own blood vessels, and that's really what we're trying to prevent. Avastin is known to target VEGF. That's important. So it's a monoclonal antibody against VEGF. And when it does that, it permeabilizes the blood-brain barrier, and it reduces tumor vascularization. So we want to say reduces here. And again, like this is true, but it's kind of secondary. And this is what is more important, that it's increasing vascularization, because that's what we're really trying to target and prevent. Here, this person has said that Avastin is a monoclonal antibody. That's important, but that is something that I would probably put here when you first introduce it. Then you don't need to put it here, and you can just continue with what it has been found to do in GBM. So we can say that it's led to progression-free survival despite normal overall survival rates. And perhaps something that would be useful to do is to take a second to explain what PFS is. So this is survival without symptoms. So patients will not experience symptoms, but they still die at the same time as with normal therapies. So it's an important thing to say, and it does improve quality of life, as this person has said, but it still has not helped overall survival. And that is what they've said here. The effects of Avastin are still short-term, and most tumors come back after 3 to 5 months of progression-free survival. So it's increasing progression-free survival by 3 to 5 months, but the overall survival is the same. And this is thought to be because of the resistance. So this is a pretty good overview of Avastin, with just a few corrections, the biggest one being that we need to promote the blood vessel aspect and not focus so much on the blood-brain barrier. So this is an edited version of the same thing. Like I said, it's great that you included the generic here. This year needs to be corrected. And then we really want to focus on the blood supply aspect and not so much on the blood-brain barrier aspect. We want to put the fact that it's a monoclonal antibody up front so that it's clear what it is, and then say that it targets VEGF. And then we want to talk more about what VEGF does. So targeting VEGF causes permeabilization, and it causes a decreased tumor vascularization. Now, if we look at the clinical trials, we found that Avastin causes an increase in progression-free survival. When we talk about survival, we want to say increase, not stretch. And it's for patients, not for the tumor. It's survival in patients. The tumor survival is obviously, the tumor is dying, so the tumor survival is decreasing, but patient survival is increasing. And then we also discussed how there's normal overall survival, even though the progression-free survival has increased. And then finally, we talk about the effects of Avastin and how they're still short-term and how the tumors still progress. And that's a great statement of what the problem is, that there's resistance, that the overall survival is not changing. And so this is something we want to think about further. Okay, so now let's workshop the final piece of the introduction, which is the introduction to CRISPR. So this person wrote that CRISPR is a highly specific and efficient tool for genome editing, that it allows researchers to alter DNA and modify gene function, and that it can function in a genome screening technology. I think my biggest thing here is that this needs to be more detailed. You saw that our other paragraphs were far more detailed in terms of introducing the technology. And so although this says what CRISPR is, I think I also need how it works here. And then you also need, after that, something, you perhaps don't need this because it sort of rephrases what you've already said, but you need more information on why it's good for a large-scale screen. So why CRISPR as opposed to anything else? And why is the functional aspect useful? So if we move to a more edited version, you want to highlight novelty. This is a really good place to highlight novelty. So anytime you write a project proposal, you want to highlight where it's new and why it's interesting. And CRISPR is by default new and interesting. And so you should highlight that for the reader. So here we've added novel, and we've added a little more detail on exactly what it is. And then we've added how it works. So the two components that guide the system and make a cut. And then we've added a little more detail on why it's good for large-scale screens, because it's simple, it's efficient, and we can multiplex it, so we can use multiple knockouts at one time. And then finally, we talk about how this high-throughput screening technology would allow us to identify specific genes and pathways, which is what we're trying to do. So we want to make sure to introduce that concept up front so that the reader knows that this is how we're going to do what we're proposing to do. Okay, so that's our introductions. Now we're going to talk about the hypothesis statement. So this goes at the very end of your introduction. And this really needs to be a strong statement because this is often what people will look at when they first look at your grant. So it must be strong. And in your hypothesis, you really want to say three things. You want to first, once again, highlight what exactly is the gap. What is our unmet need? What do we need to make sure to address? You want to follow that up with a quick sentence on any existing data. So for Avastin, for understanding why it's not working, there's really not a lot of data out there, but there are some opinions that the cells are becoming resistant over time, just like they do to other therapies. So that's probably what we would cite as the thought. We would say that there are expert opinions or papers that suggest that perhaps this is what is happening. And then you want to make one sentence that brings together all of these things. So this is sort of a very impactful sentence. It should be said as a clear statement, and it needs to bring together all three things, even if you've already said them, because this is for the reader to summarize what is your grant all about. So here we've sort of highlighted the question by saying, given the development of resistance to Avastin over time, so that sort of recaps our question that we're interested in resistance to Avastin. We then talk about applying a whole genome CRISPR-Cas9 screen to identify genes and pathways. So again, that is our hypothesis, basically, that if we apply this whole genome screen, we will be able to identify genes and pathways. And so we've said that. And then we also say it further allows us to identify novel therapeutic targets for this disease. And so that's kind of the impact, a very brief impact statement about why this is important and why it's worth doing. So altogether, this would tell the reader exactly what we're going to do in one sentence, one, succinct, strong sentence. And you really want this to be at the end of your introduction so that anybody who's confused about what's going on can just look at it and know exactly what you're trying to get at. Okay, so this is an example of a hypothesis. So this person said, through this whole genome screening, we would expect to identify essential genes involved in Avastin resistance and validate them through previous clinical data, pathway analysis, and in vitro experiments. So I would say that you really just need to say your hypothesis here. You don't need to give, this is more of like a plan, and you don't need to give a plan here. You just need to say what it is that you think you're going to do. This is a great impact statement. This last sentence is a really good impact statement. I would say the only thing that's missing is to add a little bit about the problem. So to say something about Avastin and resistance and why that's something we need to address. So this is an edited version of the same thing. And like I said, we need to add just a little bit about the problem. And then all of this is pretty good. And then we want to get rid of the plan. We don't need this plan here, so we've gotten rid of it. And we have our nice impact statement. All right, so let's talk about preliminary data really quick. So the rules of preliminary data is that it has to be your own data that you produced in the lab. You cannot cite other papers here. You could cite other papers in your section about existing data or things that are known in the field, but this has to be stuff that you have generated in the lab by yourself. And it should be clear how it adds to the grant, and it should be referenced in the text. Don't want to add things that are extraneous or not clearly important. You want to make sure it's very clear why this data is important. So some things you might add are like any data you have on Avastin resistance in GBM in the lab. Maybe you have some sort of in vivo or in vitro experiment that shows something about that. You could show anything describing the screen if you've already started the screen in your lab. So you could explain the library, how the cells are doing, any sort of preliminary analysis you have. So usually these are things like pilot studies or proof of concept studies that just show that you have the necessary resources and that you have the setup you would need to actually accomplish this project. Okay, so now let's talk about the project description piece. So as we said before, that's usually divided into 2 to 3 aims. So these are major sub-questions that the project would logically be divided into. So for example, if we were proposing doing this screen to identify specific genes, we might say that our first aim is to perform the actual screen. The second aim would be to validate any targets that we find. And the third aim would be to figure out a mechanism for maybe 1 or 2 of the targets. This is just an example, but you can set it up in any way. You could set it up to be a smaller scale where maybe, your first aim is just to do the screen and your second aim is to identify targets for a really small, like 6-week grant or 8-week grant or something. Or it could be that you've already done the screen and that's your preliminary data. And so now your aims are more like choose targets and validate them. Again, for like if you were writing a summer grant or something. So it really depends on your specific project and the amount of time you have, but this is just an example of how you might divide it up. And then for each aim, you want to make sure that you explain the question that aim is addressing and how the question contributes to the project. You also want to clearly say the methods and experiments you're going to use to address that question. And finally, you want to explain the expected results and how they're going to contribute and give a timeline. So this is an example of a project description. So this person has said that a moderately new and effective technique is CRISPR-Cas9 whole genome screening in cells. So for this first sentence, I don't think you want to highlight novelty. So you wouldn't say moderately, you would just say a novel technique is CRISPR-Cas9 screening. When you say moderately, it makes it sound like it's not that interesting, it's not that new, it's not worth doing. So you really want to highlight how good it is and be confident in your language. I would also say specifically what cell line you're going to use. They then said this would lead to the knockdown of all the genes in the genome, one at a time in parallel, while exposing cells to Avastin for 14 to 21 days. So this is in a way too detailed and in a way not detailed enough. So when you talk about it would lead to the knockdown of all the genes in the genome, that's a little too much explanation. You don't really need to say all of that, especially not the one at a time part. You would just say, I'm going to do a whole genome knockout screen. And that would be sufficient to say that you were going to knock down every single gene in the genome. You do want to say which cell line, but you don't want to say preferably because it implies that you don't know what you're going to use. And whenever you're writing a grant, you want to be really clear about your experimental setup. So you should just say we're going to use SNV19 cells, or we're going to use XX cell line because that's what we have in the lab, or because that's what's best for this type of project. You should have a reason for the cell line you're going to choose, and you should be really clear about which one it is. And then again, with the time period, it's the same commentary. You want to make sure that you're just really clear. So you pick 14 or you pick 21, but you don't say both. I would probably choose 14 in this case, so we would say 14 days. When you're writing a grant, you don't want to do this kind of thing, so no slashes. You would just say expressed or knocked down or something like that, but you wouldn't put a slash like this. And then you want to really explain how you know this. So how are you going to do this analysis, and how does the screen tell you what you think it's going to tell you? So I think my biggest gripe with this overall is that it's not detailed enough. An aim paragraph needs to be much, much longer than this, because if there's one place in your grant to be detailed, it's where your aims are, because this is what people are going to look at to know that you know what you're doing and how to accomplish this project. So it should really detail your entire experiment, how it's going to work, exactly how you're going to do it, and what results you're going to get. There is some nuance. It doesn't need to be so detailed that you would go through the entire methodology of the experiment. So you wouldn't tell the person that I'm going to take 1 million cells and infect them like this and add exactly this much virus and then plate them on these many plates. That's too much detail. That's methodology. But what you would say is I'm going to use this technology with this cell line and this treatment for this many days, and I'm going to analyze it like this. So you need to make sure it's very clear what the setup is, but you don't need to go into how to do commonly known experiments. So this is a more detailed version of the same thing from someone else. So this is more what it should look like. It should be a fairly detailed, pretty long paragraph. Often for longer grants, an aim is an entire page or even more. So let's go through this one. So here we're talking about a pilot whole genome CRISPR screen. So it's not really going to be a pilot, it's just going to be a whole genome CRISPR screen. In immortalized recurrent GBM cells. So again, the same comment. You need to make sure to say which cells. And I wouldn't say things like have been previously treated and become resistant, because that's almost a project in itself to establish a resistant cell line. So you should choose a cell line that has already been established, that people know about, that you can obtain, or a cell line that already exists in your lab that you can just explain. Otherwise, this becomes a whole nother aim to establish the cell line and figure out if it's resistant and characterize it, and then you could continue with your screen. This person then said assess an Avastin treated population in comparison with a controlled untreated population, which is perfect. They talked about their screen setup, and then they talked about the exact treatments and the doses and the days, which is very appropriate. The only change I would make is that this is an animal dose, and when we treat cells, we talk about micromolar doses, so you would want to adjust that. They then talked about the analysis, so all of this is talking about analysis, and this is great. My only commentary here is that this could probably be consolidated just a little bit, but it's very detailed and very clear about how the analysis will work, and that's important, because you want to make sure that your readers understand that you know how to analyze your data and that you know what you're going to get out of it. And then at the end, instead of saying it is hypothesized, I would just make a very declarative statement of like, I think, or I know, or this is what I will get out of it, that this screen is going to allow me to identify networks and genes that contribute to resistance. And that from this is how I'll move into my next aim to figure out which targets to use. So this is a workshopped version of the same thing. So as we talked about, we don't want to say pilot. We talk about the whole genome screen. We specify a cell line. And we talk about the treatments we're going to do and exactly how the screen will work. So This is all perfect. The exact number of days, the treatments, the doses. You want to make sure to include all of that because that describes your setup. And then you're going to talk about the analysis. So again, here, I think this could be consolidated. I didn't actually consolidate it, but you could probably reword it to be still as clear, but a little bit shorter. And then we want to make this part more declarative as just a summary statement of what this aim is going to show once we complete this experiment and why it's important for our project overall. And then we want to talk about the impact statement for the project overall. And then in our next paragraph, which will be our next aim, we can talk about the target identification and validation. But overall, this is a great example because it clearly goes through what your question is here, then clearly goes through all of the experimental methodology needed. So sometimes for some aims, you're going to need multiple experiments, but in this case, our aim is to complete a CRISPR screen, which is a large experiment in itself, so it's just one experiment. It clearly explains all the different setup components within that experiment, clearly explains the analysis, and then goes through how this experiment is going to add to our project overall. The only final thing that you might add is a timeline. So perhaps doing a screen like this would take 2 months. So now let's talk about the significance statement. So this is the final piece of your project proposal. And like we talked about before, it really needs to talk about why this project is important. And in GBM, this is kind of already set up for you because we know it's fatal. We know we need new targets. We need novel approaches. We need new drugs. And all of these things are great reasons for us to do any new project on GBM to try to find a better way to treat it. Other things could be that CRISPR is very novel. So it's a really novel approach that gives you lots of new targets, not just one. So it has a lot more potential to identify something that might actually work. And you can also add things about how, like, if there's a big hole in the literature, nobody has really addressed this. That's another great reason, another reason that your work would be really significant. So you just want to give every reason why your work is interesting and significant and why people should be interested, but try to keep it really succinct and hard-hitting. And this goes at the very end of your grant and should really be very strongly written. Okay, so let's say that you wrote this entire grant and you give it your best shot. This is your first draft. Who do you ask for help, or who should you ask to review this? So let's say if you're an undergrad or a medical student or an early graduate student, you'll often have an advisor who is assigned to you to help you out with such things. So this is a great first person to go to take a quick look at it and give you general comments on things to include and make sure you have all the necessary paperwork and other components. Other people who are also really helpful as a first pass are people in your lab. So grad students or postdocs that you're working with have often written multiple grants and can definitely take a look at your grant and help you out and edit it. And then once you've gone through a round or two of edits with those people, your PI is also a great resource, but usually after you've asked other people, because you want to make sure that you have something good to show your PI so that they can make meaningful edits without wasting time just editing stylistic things and other small things, but they can really give you big picture edits, and so that what you give them looks really good, and so they trust that you know what you're doing. Thank you so much for listening. If you have any questions or feedback, please feel free to reach out to us on any of these platforms.